So thanks everybody for joining. It's a pleasure today to have Charlie Khemtong, who is a chemist by training here, and he's going to talk about a topic that I think we've long talked about using radioisotopes to better understand diabetes. He's from Thailand, where he earned his Bachelor of Science, went on to get his Ph.D. from the University of Akron, and then went to a famous place for understanding diabetes that at the UT Southwestern in Dallas, where he rose to the rank of assistant professor. And now he's been here, and we just talked about this 2 years. So he is going to present, I think, a very innovative talk today. And we have many people online. And thank you, Charlie, for coming. Appreciate it. Thank you.
Thank you. Can everyone hear me? People in Zoom? Okay, well, it's a great pleasure to be talking here. One thing I want to point out is what I'm going to talk about is going to be very different from what you guys have heard in this series. So I'm going to bring in a different aspect of how to look at the metabolic questions using what I would probably want to call it a chemist perspective. So I'll try to not go too much into the technical background, but want to give an overall picture of what we're trying to do to everyone. Okay, so I'm going to start by giving you guys a little introduction of there's a link of metabolic abnormalities and substrate metabolism or substrate utilization. I mean, this is a well-known fact, obviously. And then I'll talk about how we can actually use a novel tool, which is relatively new, to actually analyze the changes in the substrate metabolism. And then I'll give you guys a few examples of the work that have been done in our lab in the past 5-6 years, looking at changes in metabolism. As we are all aware, we are blessed with metabolic flexibility both in the macroscopic scale and in a cellular scale, meaning that our bodies, our cell can utilize different substrates for ATP production, for energy production. And when we're healthy, these preferences can be switched and alternate as a needed basis. For example, you wake up, you don't have a lot of carbohydrate. Obviously, our heart, for example, is going to start using a lot of fatty acid for ATP production. When we start going out for a run, other substrate will also come into play and help provide the ATP source when the energy demand is increased. And of course, we also know that when we get sick, diabetes or fatty liver, that's going to change. The flexibility that we have would be compromised. And the main question is, how do we know that? I mean, obviously, it's not that hard to detect any changes when it's become very, very severe. But how do we actually look at these changes when it's very subtle, when it's at the early onset of the disease? For example, you can look at it here. People with non-alcoholic fatty liver, we have a decreased overall fatty acid oxidation, and you also, if you look at specifically in the liver, you can also see that. So we know that. We know there's the changes in the cells, in the tissues, and as a whole, there's changes in metabolism, there's changes in the substrate utilization. The main question is, how do we actually detect that? What kind of tools are out there? Of course, like I mentioned earlier, when it's getting really, really bad, it's not that hard to detect. But if we can actually look at that earlier, it would be very beneficial in terms of the diagnosis, treatment, and management of the disease. So I'm going to go back and talk about a very simple view of metabolism. So maybe to you guys, I would just call it my simple view, because a lot of people here are expert in the metabolism. So I look at the metabolism. What I see is chemical compounds. And I'm a chemist, obviously, going through, in this case, an enzyme through a chemical reaction. You can call this metabolism. And then those compounds or substrates or energy fields would be converted into the products. that's called metabolites. It could stop there, or it could also continue in the metabolic pathway and get used for other pathways as well. So in terms of analyzing metabolism, there are quite a few ways that we can look at this, but what I'm thinking about, let me go back and okay, sorry. What I'm thinking about is there are three major ways of coming up with a question for how to analyze metabolism. The first one is how much of a certain metabolism we have, or certain metabolite we have, because we know if you have a lot of a product of certain metabolic pathway, you would assume that that pathway is very, very active. So there's a lot of imaging modalities out there that will give you that information. In this case, example that I'm showing here is I'm still trying to use this. A relatively new and novel imaging technology is called deuterium MRI. So basically, you guys think about MRI, we actually look at water protons, which we have a lot in our body. But in this case, you actually look at deuterium. So if you replace hydrogen with deuterium, you can actually have a metabolite that contains deuterium. And in this case, they actually put in glucose that had deuterium on it in a human patient in a brain tumor. And you look at it and say, can we actually look at a lactate? Is glucose being utilized a lot in this tumor? And that's what they see. Again, in terms of asking question here, it's not a new question. It's known when you have a tumor, you have the Warburg effect, elevated aerobic glycolysis. So you can use some tools to look and see how much of certain metabolites or pathway is being is going on in a certain tissue. Another thing that we can take a look at is you see anything unusual? This is probably part of the field that's called metabolomics. So what that means is you actually look at the let's say a tissue of a healthy individual versus a tissue from an individual who has a metabolic disease, and look at it and compare metabolites, compare the molecules in there, and say, do we see anything different? Obviously, if you do see something different, that's probably a biomarker. And then you can go back and look and say how much of those we have in there. So in this example is pointed out the oncometabolite. I apologize that I have a lot of cancer examples here, but it's the same for all metabolic diseases. These two methods will be a little, when I say easy, for people who work in the field probably wouldn't agree, but it's less challenging than the third question that I'm going to show you guys is how fast. We know we want to measure a certain pathway. Is there a way to actually measure the rate, the speed? Metabolomics is you counting number of cars. A lot of people use this analogy, which may or may not be too hard if you send the people to the parking lot and count them at a certain time. But the more challenging is how fast are those cars traveling? You're going to have someone at a certain point, and then you have a radar detector to look at the speed. So this is, to me, is a more challenging question in terms of answering the metabolic questions.
Charlie, just a quick question, and again, best thing. Do these three always run together, or in different scenarios do you have permutations or abnormalities in one and not the other two, or two and not three? How distinct are these?
I would think they probably go hand in hand. Although the speed may, the flux may not. But if you have, let's say, let's say the Warburg effect, it doesn't have to be super fast, but if you have that going on for a long time, you can have an increased lactate accumulation in tissue or lactate acidosis. Okay, so of course, if you want to take a look at this, it will be simpler if we can actually trace the metabolism. So one way to trace this is to use isotopes. As you probably remember from biochemistry and chemistry courses, all of these substrates, metabolites or fields, they're all organic molecules. They have carbon, hydrogen, oxygen, sulfur, nitrogen, and all those. And all of these have different isotopes, obviously, and you can pick which one you want and which one is available. So for example, hydrogen has 3 isotopes and 2 of them are stable. The one is the hydrogen proton and then deuterium that I mentioned earlier. In carbon, there's 3 naturally occurring isotopes. One, the majority is the carbon-12, and then you also have carbon-13. And those 2 are stable isotopes, and then you have carbon-14, which is a very, very small amount, but it's very sensitive. That's what's used for carbon dating. So of course, we can use radioisotope as tracers for metabolism. In this case, this is the gold standard for metabolic imaging. Is F18 glucose, so or FDG. So what happens is you injected a glucose molecule that's been modified with the fluorinated atom that has a radioisotope activity. So when you inject it to a patient, and then these would travel and fade, kind of mimic the dose of glucose. Although the main problem that FDG has is it doesn't get metabolized. Obviously, you guys can imagine the enzymes know that this is not glucose. It's just modified glucose. So when it's get into the cells via the transporter and get through hexokinase, and then they'll stop. Although it's still giving us a lot of good information of what happened to glucose metabolism or uptake. Recently, there's a lot of interest in developing a PET tracers that has carbon-11. This is not a natural isotope, but you can actually synthesize this. When you introduce carbon-11, the enzymes, even though it has a slightly different mass compared to a naturally occurring carbon-13 and 12, the enzyme will still treat these isotopes as the same molecule. So we get through the metabolic reactions that I mentioned earlier. So in this case, you see C11 palmitate and then C11 acetate. But this is not to knock down PET field as a whole, but PET tracers or PET uptake relies on the internalization of these molecules, and a lot of the time it doesn't really give us the whole picture of metabolism. So one being that FDG doesn't really get metabolized throughout the whole glycolysis pathway. But once you use carbon-11, the molecules get metabolized. But the problem is when you detect them, it doesn't matter what PET tracer you use, you're still detecting the same particle. So what that means is even if you have the molecule of your substrate or the molecules of your product, you're still seeing the same signal. You can't differentiate and say, oh yeah, when glucose, C11 glucose get uptaken into the cells, and then it becomes acetyl-CoA, lactate, or glutamate, you won't be able to tell because it's giving you the same molecule. So that's exactly what we have here. So what our lab is, again, I'm not taking credit for this. We came in when the field was already pretty quite established. The question is, what about other isotopes of carbon or hydrogen or something like that? But our lab is focused on carbon. For example, stable isotopes. Instead of detecting radionuclide that's being produced from radioisotopes, can we actually look at stable isotope like carbon-13, and actually, instead of looking at the same particle, we actually look at different species of carbon-13 metabolites. So I did some literature search and see how far back did carbon-13 tracers go in the history. I'm not sure if I found the original one, but this seems to be pretty old, 75 years ago almost. So this is an editorial in Nature. It's just like a lot of things when we talk about some ideas and you just keep ideas to other people. So again, I saw that this tracer has quote unquote on it. So I wasn't sure if this is the first time the word tracer was coined, but I'll do some more research here. So what this says here is if you have the substrates that's labeled with C-13, and emphasizing the C-13 and C-12 are pretty much the same chemically. potentially can use this as a tracer to follow the history of carbon atoms in their passage through biochemical reactions. And an interesting thing here is, this is an example that even during that time, even small quantity of carbon-13 enriched sugar is eaten by or injected into an animal. The distribution and state of chemical combination of the carbon can be determined by the ratio of carbon-13 and carbon-12. That will tell you about what happened to the carbon-13 label of glucose. So exactly, if you put that description into a schematic presentation, you will see here, you can have glucose level with carbon-13. You get through a glycolysis and give you a 2-molecule pyruvate, which can go to lactate. And pyruvate can get into the TCA cycle, being oxidized into acetyl-CoA and 1 molecule of CO2. And acetyl-CoA gets into the TCA cycle and continue the journey in becoming a TCA cycle metabolite. Can we actually trace things? And the question is, we can, and people have shown it, and this is, to me, I think it's quite exciting field of research. So here it was the work that was done by a group back at UT Southwestern, Kevin Cordy, Raf Badinis, and Bob Baku, Elizabeth Marr. So what they did was they infused carbon-13 labeled glucose into patients with GBM, glioblastoma, and clear cell renal cell carcinoma, and take a look and see what happened what happened with the metabolism of glucose in these patients in these tissues. If you want to take a look at this, this is the filled circles. Blue represent carbon-13, and the open ones represent naturally occurring carbon-12. You can take a look at any step of the metabolism and analyze and answer one of those questions that I mentioned earlier. How much of certain metabolites it is, and how fast. We can actually calculate flux with a lot of fitting models that are out there. So what they found here is carbohydrate oxidation into the TCA cycle via acetyl-CoA is unexpectedly preserved in lung cancer and intracranial malignancies. That's what they found. This is not what we would expect because of the Warburg effect, obviously. But in the clear cell renal cell carcinoma, they found that the glucose oxidation into the TCA cycle is decreased. And again, this is not something unusual, because this follows the Warburg effect exactly. So the way this guy,
yeah, I had a question about whether you have immunologically hot tumors versus not, and if you see any immune metabolism in these situations.
That could be, but I can't really answer that question because I don't, we probably can go back and take a look at the paper, and there might be some answer for that. But it could be. I mean, of course, the tumors that might be hotter or more active than others. So, yeah.
And follow. Can you go back to slide, please? Oops, back. Yeah.
Oh, how do I go back? Okay.
Sorry.
That's all right. Okay.
Right. So I'm trying to figure this out here. So if you come in on a front load, and you would think that normally you would have degradation as you go throughout this pathway, or utilization, whatever you want to call it.
Mm-hmm.
If there's a major fall off along this pathway, you would be able to find. That's the major notion.
Yeah.
How do you separate out? I guess where are you going to identify inflection points, or gosh, whatever I say, major fall off or whatever.
So in this case, they just look at they use mass spec to look at it. So they wanted to see the Warburg effect, meaning that they want to see more lactate whether or not lactate over the ratio of citrate, which reflects the incorporation of these C-13 compounds into the TCA cycle. As you can see here, citrate right here. So where's the fall off?
Let me rephrase the question. So if you have 100% input, the notion is that at the end of the day, you can get 100% output. The notion here that somewhere along the way in this pathway, if there's a blowout, if you would, it's a flat tire, you would be able to identify this.
Yes, actually, I think I got it. So for example, if these were to go over and over, around and around the TCA cycle, what if there's something wrong in a certain reaction, right? Like, then that step won't go forward. So we will probably see, let's say, for example, here, if there's a mutation in the enzyme to convert succinate to fumarate, for example, and then we'll have a lot of carbon-13 label in the succinate, and we won't see a lot of it at all in the fumarate. So if we see that, then we can say that's a mutation or that's an abnormal metabolism from succinate to malate. So we can actually look at that. I hope I answered your question. Okay. So, question.
So I have a question with regards to conditions like obesity and diabetes, when there is insulin resistance or lack of insulin production, that leads to reduced glucose uptake. So how do these ratios work?
If it reduces glucose uptake and glucose oxidation, and then we won't see, we can go back. Now I know it's a different button. We can go back, and again, this is cancer, but we can still use the same diagram. You have glucose going in. In this case, they choose to analyze citrate. You know, how much citrate is being labeled with carbon-13. We can also use glutamate, which is what we do in our lab. But you look at, let's say you want to look at the liver biopsy of people with diabetes or fatty liver. You take a biopsy and you can take a look and see how much of the citrate that you have from each tissue is being labeled with carbon-13. And if you have a lot of glucose oxidation into the TCA cycle, you will see more citrate with carbon-13 label, right? If it's not being oxidized a lot, then you won't see a lot. But that's just an easy example to answer your question, because you know you have a lot of other factors that will come in and say, you know, what time of the day are you fast, are you fit, stuff like that. Okay, so this is quite a very difficult topic here, to be honest, and you know, even my students take a long time to get it. It's hard to really convey this message in a short talk. So you can use mass spec, obviously, but our lab is very interested in the magnetic resonance in the form of NMR, nuclear magnetic resonance, or magnetic resonance imaging MRI. That is because of the potential to actually do any of these tests in vivo in human patients, obviously, without having to non-invasive, without having to invasively take out the tissues. The good thing is carbon-13 is magnetic, so they're just tiny magnets, and you can actually detect them using these two technologies that I talked about. So, but the question is, can we actually detect carbon-13 like what I showed you in the first few slides of deuterium? Is carbon-13 sensitive enough? The answer is no. Carbon-13 is not very sensitive, and on top of that, you have only 1.1% naturally occurring in any substrate you look at. So we will need to add, we will need to increase that. You know, obviously, we will do the isotope enrichment, which luckily is not very hard. There's some chemists out there in the world who can do that, and they sell them to you. that will take care of how much carbon-13 you have. But in terms of not sensitive, you can't really use MRI to detect carbon-13 because physically, not physically, in terms of physics, these magnets are just not very sensitive. So we have to find a way to what we call hyperpolarized them. Actually, I know that people in certain field will actually perceive the term hyperpolarized differently than what we use here. Anyway, so this is just a cartoon. Don't worry about the physics of it. The reason why MRI and NMR is very insensitive is because when we think about magnet like carbon-13 or proton in the MRI, for example, when you're looking at is how they align themselves along the magnetic field. So you think about putting these magnets into an MRI, which is a giant magnet, and these little magnets will either align along the magnetic field or against. And what you're looking at is to produce a signal, you just have to look at the difference between the two states of lining against versus lining along the magnetic field, which is not a very sensitive way. So luckily, about 15 years ago, there was a first report of the technique called dynamic nuclear polarization, which would put metabolic substrates in a magnet, very high magnetic instrument called polarizer, and phase it to almost absolute zero. So in this case, it's about 0.2 or 1 Kelvin, depends on what system you have. And then what that would do is the difference between lining against and lining along the magnetic field will be huge. So you can actually look at it. So that's this is an example of a very first polarizer that was used here. So these are the pictures of the polarizer that I mentioned. This was back at UT Southwestern before I moved here. The one on the left is the preclinical polarizer. You put this, you get the solution. You cannot inject into the human patients. You can do it in a test tube or animals. But the one on the right is pretty much the same, just better looking, or it's more than just better looking. It's just you have GMP and quality control unit next to it. That is for human studies setup. So you have a solution of your metabolic substrate putting into inside of this magnet, let it polarize for about 2-3 hours, bring it up, shoot hot water into it. You get a solution at 37 degrees Celsius. It depends on what study you're doing. You can inject it into what we call an NMR spectrometer, will give you peaks, and that's what we do a lot. And you can also inject this solution into animal or human patients, depends on which scanner you use. So with that in mind, the first study that people did was to see glucose metabolism. Obviously, we know carbohydrate metabolism is changing quite a lot in a lot of disease. So the first study they did is to see whether or not they can actually look at the Warburg effect in cancer. So what they had was they had a carbon-13 label on enriched glucose, polarize it, and inject into tumor-bearing mice. And as you can see here, this is the hot ROI here. This is the injected molecules of glucose, and you can see the outline of the tumor. And you look at the lactate, which is the downstream metabolite of glucose, reflecting glycolysis. You can see that in the tumor it's hyper intense, whereas in other tissue you don't see it. So this is just a preliminary study to show that glucose metabolism can be monitored, can be detected in real time using hyperpolarized C-13 or carbon-13 MRI. One thing about glucose is because of MR physics. Whatever signal that you have, hyperpolarized C-13 MR signal that you have doesn't last very long. So there's no way that C-13 glucose can be used in human patients because of the limitation that I mentioned earlier. So you won't be able to have time to check whether or not the pH is good, the solution is suitable for injection, for example. So the most ideal surrogate for glucose is pyruvate. And as we all know, one glucose molecule gives you 2 molecules of pyruvate. The good thing about pyruvate is it has 2 carbonyl or carboxyl, 1 carbonyl and 1 carboxyl group. And both of them are very favorable in terms of C-13 hyperpolarized MR properties. And when you use these, you can polarize it and take a look here. When you polarize it, inject into the bloodstream, it will flow through and then get into the cells and get transported and get oxidized, get metabolized to 3 different directions. One is being into lactate that we talked about earlier, and it can also be metabolized to alanine. Glucose can also, pyruvate can also be metabolized into the TCA cycle, forming acetyl-CoA. It depends on where you put the carbon-13 atom in. You can actually look and see if you want to see CO2, which is cleaved off the pyruvate molecule by pyruvate dehydrogenase enzyme, or you want to look at the TCA cycle intermediate. And obviously, the first example of this is to look at a tumor, because again, I apologize for having all tumors, because this field was actually developed for cancer in the beginning, I guess, but now it's been applied in other metabolic diseases as well. So this is to look at how much of lactate is being converted from a hyperpolarized C-13 pyruvate. And this is to see whether or not you have the Warburg effect, and that's actually what they found. So those are the introduction and examples of some of the work that were pioneers in the field. And going forward, I'm going to show you guys examples of the work that we've done in the lab. Since moving to UF just a little over 2 years ago, we've been trying to continue the work that we did back in Dallas, involving the use of carbon-13 tracers. Most of the work that we do, we use hyperpolarized C-13 MR, that being NMR and MRI, to look at the metabolism of metabolic substrate here. And as a chemist, I also have a little, I won't say a little project, we actually got funded with an R1. We also have a project that develops a new imaging agent for measuring tissue pH in animals. And the third part of the work that's going on in our lab is to just trace metabolic activities or probes in isolated perfused tissues in animals, and eventually we're going to try to do this in patients as well. Although this is something that I'm not really going to focus much on today, but hopefully I'll be able to show that to you guys later on. So the first example that I want to show you is how we use hyperpolarized C-13 NMR to actually detect a rapid glycogen mobilization in the heart. This is stimulated by isoproterenol in perfused red heart. So the way that we did this study is we isolated the heart from healthy rats, and then we perfused it into our using our perfusion setup. It's not exactly this one, but it's the same concept. And then we put the whole thing into an NMR spectrometer. I keep putting it here. And then when we're ready, we inject hyperpolarized C-13, in this case, pyruvate, into the heart and detect and follow what happened with hyperpolarized C-13 pyruvate. So this is an isolated perfused rat heart that we do in our lab. So we set out to actually answer and see when you stimulate the heart with isoproterenol or any catecholamine, the heart will work harder and oxygen consumption will increase, energy demand will increase. We wanted to see if oxidation of hyperpolarized C-13 pyruvate will increase, and if we can actually detect it. So we perfuse it and then inject this isoproterenol and then the hyperpolarized C-13 pyruvate. So that's what we wanted to see is pyruvate being oxidized into the TCA cycle, and can we actually detect the oxidation? So what we saw is actually the exact opposite of not exact opposite. That's not what we wanted to see. It was the production of CO2 from the oxidation of pyruvate never increased regardless of how many times we stimulated heart with isoproterenol. But what we saw was the production of lactate. As you can see here in the control heart without isoproterenol, lactate will increase from the arrival of hyperpolarized C-13 pyruvate, and eventually is dying down. That is the hyperpolarized signal. It's not because lactate is not being produced. But what we saw is in the heart that we stimulated with isoproterenol, you can see the second increase in the lactate signal, and it buffers us for a while and see what happened. Why is it a lactate increase? We did a lot of other studies by using non-hyperpolarized C-13 studies. We just injected the heart with carbon-13 pyruvate labeled glycogen, and that was one of the last experiments we did. What we found was when you stimulate the heart with isoproterenol, the energy demand is increased so much, and being an isolated perfused setup, you don't have circulating nutrients provided to the heart. So the heart resorted to mobilizing glycogen to produce sugar. So what we have is we have a rapid breakdown of glycogen, producing a lot of lactate, and is one of them, obviously. And hyperpolarized C-13 pyruvate that we injected actually mixed with the new lactate that comes from glycogen, and therefore we have a large signal increase in the lactate. So that is something that we were very excited, very proud of. We have a technique that you can actually look at a rapid metabolic changes. All of this happened within about 10 seconds, and it gives us the spatial resolution, the temporal resolution that we can actually look at these metabolisms. We have also looked at the metabolism of pyruvate in isolated perfused kidneys. The reason is being because there's a lot of interest in imaging metabolism in kidneys, obviously. But the main question for people working in this field is when you image the kidney where everything goes and be secreted out, are you detecting the metabolism that's being produced in the kidneys, or you are detecting metabolites that being produced somewhere else and being delivered there via excretion? So that's why we were interested in this. So we did similar experiment that we did with the heart. We isolated the kidney, cannulated and perfused it with a mixture of pyruvate and octanoate, a competing substrate, or just pyruvate, and see what happens. Is the kidney being metabolically active and utilizing pyruvate? So what we found is the kidney is actually very, very oxidative. It oxidizes a lot of pyruvate and also octanoate, although octanoate is not hyperpolarized, so we can't see octanoate metabolism. We can only see pyruvate. So that's by design, because we wanted to see pyruvate. So if you look at the signal here, bicarbonate, which is equilibrated with the CO2 and has a higher signal, we look at it as an indicator marker for pyruvate oxidation. You can see when you don't have octanoate for the kidney, obviously pyruvate is being oxidized to a high degree than when you have a competing substrate. Again, in terms of metabolism, we're not really answering a challenging question. or anything. But in terms of technology, this is something that's very exciting. You can actually detect the substrate competition, substrate utilization in a viable organ. So these results were confirmed by taking out the tissues after we finished the experiment and run high resolution NMR and calculate flux. And you can see the flux through pyruvate dehydrogenase is actually higher when you have on fire. Again, that's to be expected. One thing that we found is very interesting is that unlike the heart, if you have a pyruvate, you give the heart pyruvate, and then you have a substrate, a competing substrate, in this case, alkanoate, which is fat, the heart love fat. It's going to switch completely to alkanoate, so you won't see pyruvate oxidation. This is the work that was reported by our very own Dr. Mary in the biochemistry. This again was done back at UT Southwestern. But in the kidney, we don't see that. We see that when you have alkanoate, there's only a decrease in pyruvate oxidation. It doesn't abolish that pathway. That's because the kidney can utilize multiple substrates and prefers to rely on multiple substrates instead of sole substrate, like what happens in the heart in this case. This is probably going to be more relevant to what many of the audience are working on. So this is probably more like metabolic modeling that we have shown so far. This is the study to look at pyruvate oxidation in diet-induced obese mouse livers. And we have a model of knockout of pyruvate dehydrogenase kinase, isoform 2 and 4. This was provided to us by our collaborators back in Dallas, Augustine Chuan. When you knock out or inhibit PDK, obviously PDK inhibits the pyruvate dehydrogenase or oxidation of pyruvate. So when you knock out or inhibit this enzyme PDK, PDH is going to be activated. So what you're going to see is an increased oxidation of glucose and pyruvate in this case. So what we saw is obviously when you have an obese liver or a liver from an obese mouse, pyruvate oxidation or glucose oxidation is decreased. But when you knock out these two PDK isoforms, you will restore to some extent the oxidation of pyruvate oxidation. And we've seen this in vivo in real time, as well as looking at the tissue extract after the fact, and they confirm. So an interesting thing about this is potentially we probably can use this and say, if a patient comes in, you have something wrong with the liver, we can actually detect say glucose oxidation after 6 months or a year of treatment. We do that exam again. Do we see any change? And that's just a simple view of what we're trying to do in terms of the translational aspect of this. Okay, so that just to summarize this part, I have probably have to go through the latter part a little quickly. Is that hyperpolarized C-13 NMR or MRI can actually reveal what happened in terms of substrate oxidation, substrate utilization in real time. Again, this is something that it's not easy to do. We know that we change, but can we actually detect it? So I'll switch gear a little bit and talk about how to use carbon-13 MR to actually measure cellular homeostasis. The first one I'm going to talk about is cellular redox. Some of us probably remember that the mitochondrial dysfunction is present in many metabolic diseases, and one of the outcome of that is you have an accumulation of NADH, the reduced form, and a redox of the mitochondria is not going to be normal. Again, measuring redox is not an easy task, be it in vivo or even in cells, because a lot of time people resort to measuring NAD and NADH intake ratio, or another aspect that people use is to look at the redox pairs. If you want to look at the mitochondria, you look at ketones, or acetoacetate is oxidized form, and beta-hydroxybutyrate is the reduced form. When the redox chain takes place in mitochondria, the ratio of these two molecules would be changed as well. So in our lab, instead of measuring this, we wanted to see if we can actually detect the changes in real time. So we use carbon-13 label acetoacetate, and we hyperpolarize them. You guys probably can see the diagram here, where you inject the acetoacetate again to the cell in the mitochondrion, and then gets reduced to beta-hydroxybutyrate by enzyme beta-hydroxybutyrate dehydrogenase. And we have a lot of NADH accumulation in the mitochondria. You would expect to see a high production of beta-hydroxybutyrate. We did the experiment using the setup that we know very well, isolated perfused heart, taking the heart out from a rat and perfuse them under the normal perfusion pressure, and compare this with the low flow ischemia. This is something that can happen when you have a blockage in the artery. And we also treated the heart with rotenone, which is a mitochondrial complex 1 inhibitor. This is positive control to show that if you treat the heart with rotenone, you're going to see a lot of NADH accumulation, potentially the increased production of beta-hydroxybutyrate. And that's exactly what we saw here. Again, this is the time course of the signal of beta-hydroxybutyrate. Visually, you can see that when we have the ischemia or rotenone, the signal is increasing at a much greater degree than when you look at the control heart. And you take the area under the curve. This is to quantify and see how much we have in total. This is, it's easier to take a look and compare. You can see that when the heart is undergoing low flow ischemia, you can actually see an increase in production of beta-hydroxybutyrate. So we compare this and look at the mitochondria in these hearts and see if the morphology and the function of mitochondria are still okay. Do they actually correspond to what we see in terms of beta-hydroxybutyrate signals? And we saw that obviously when you induce global ischemia, low flow ischemia, or rotenone, mitochondria are very sick, are very dysfunctional. And that corresponds well with the level of beta-hydroxybutyrate signal that we see. And we also look at the NAD/NADH ratio, and these are all agreed. So why is this important? It's important because potentially we can use this when we translate into the in vivo application. We can use this technology to actually assess the health and conditions of the mitochondria in the heart. And this is something that we are trying to pursue forward and hopefully wrap up a grant application in the near future.
Sure.
Yes, sir.
So you seem to flow so easily from heart to kidney to liver, but I'm not understanding why you flow so liberally through those, which is fine. But and then why is that? And do you see correlation between the different organs if you're looking for dysfunction or ischemia? Or some of these organ specific, why are you flowing like this?
So that's a good point. When I started working on this, I was more of an imaging person. So we have a tool. I mean, that sounds kind of bad. We have a tool. We want to find the question. It's true that you see the changes in different organs. But back in the days, I'm talking about 10 years ago, we didn't know. As a field, we didn't know. For example, in the kidney, that was the first paper that was shown that you can actually look at it. So there are a group of people back at UCSF in the UK and Dallas as well, they do this in human studies, a similar thing. But the one thing to jumping into the human studies is, just like I mentioned earlier, you see the metabolites and you see the marker. One of the questions we have is, are those markers being produced there or being delivered from other organs? So that's why we did isolated organ. The reason why we switch organ is because we wanted to see, if we can actually take a look at this in different organs and do the work, for example. For the liver, actually, our collaborator actually approached us and say, hey, we have this model, can you actually look at that? And that's it.
I still don't understand the answer to the question. Do you see correlation across organs in terms of your outcome, or are these specific for tissue?
Do we see correlation, or we do see correlation? No, I'm asking. Well, we can actually talk about it after this.
Okay.
Yeah. Okay. We also expanded this work and look and see if we can actually measure the redox state or the redox changes both in the cytosolic compartment, which is something we haven't done before, and in the mitochondrial compartment at the same time. So what we did is we used two probes, hyperpolarized C-13 Pyruvate, with it following the reduction of pyruvate to lactate, similar to reduction of acetoacetate to beta-hydroxybutyrate, and look at it. And what we found is under the ischemia and rotenone treatment, the appearance of beta-hydroxybutyrate agrees pretty well with the redox state of the mitochondria. In the beginning, we were surprised by this, but what we found was the appearance of lactate being produced from pyruvate does not really correspond to the changes in the cytosolic redox changes. You do see the trend, obviously, but we did, again, the number is still quite small. But what we found is it's not really correlating very well. So this doesn't mean that the cytosolic redox doesn't change. It just means that pyruvate to lactate may not be the best marker for quantifying or measuring the cytosolic redox. But again, the number of hearts here is a little bit small, so more studies need to be done here. So I'll skip the last part because that's we're running out of time, and I would want to take this opportunity to answer any questions that you guys have. Thank you very much.
So from my perspective,
Thank you.
so much of this is going over my head, so I'll sort of say that right up front. I'm sort of reading about your talk. I don't know much about it, but I'd heard about people using deuterium.
Yeah.
In terms of MRI, do you use deuterium? Is there any superiority of using deuterium versus carbon-13? And that was the question.
Well, that's pros and cons. Deuterium, when I say carbon-13 is not sensitive, but we can actually polarize. Deuterium is not sensitive. So you have to inject, let's say with a patient, you have to ask them to drink or inject a lot of glucose. For glucose, it's probably okay, but some other substrates probably not. And then when you image it, you have to image in a high field, meaning that you have to use a magnet that's very strong, 7T, which is not approved for clinical uses routinely, although you can do human studies in them, and 9.4T, which is something that's bigger. But in terms of technology, it's simpler. The people who actually did the imaging probably won't agree, but you have deuterated substrate. You infuse it into the patient, and you just let them lay there, and then just acquire over and over, and you get a map. So it's simpler. For hyperpolarized C-13 MRI, it's a little more complicated. You have a polarizer, and then I know I mentioned a few times that you don't have a signal that lasts forever. So you polarize it, and then it's going to come down to a thermal equilibrium state, depending on the NMR physics of the molecule that we're looking at. But with carbon-13, as you can see, you can actually take a data point in every second or two. Our study, we did it every 1 second. That gives you temporal resolution. You can actually see a real rate. With deuterium, most of the time you probably see accumulation, which is not a bad thing, but it will probably answer different questions, I guess.
So I guess I have a question. You keep saying it's very sensitive, but I'm trying to understand where that threshold is. What is sort of the minimal cell numbers that are highly metabolically active that you can signal over background?
So in terms of MRI, I don't think we can actually define that, because MRI itself is not, when I say sensitive periods, it's spatial sensitivity. When cells are close, you can't tell this cell is this, that cell is that. So when I say sensitive is to compare with MRI. But in terms of coming up with a cellular number, cell number threshold, I don't think MRI can do that as a whole, not even hyperpolarized.
Yeah, I mean, we come at this sort of with a bias of an interest in maybe immune inflammation or tissue inflammation. And I guess my question would be, how much Warburg metabolism do you need to see to be able to detect that? Would it be an inflamed lymph node? Would it be infiltrate? I guess.
Yeah, I don't think I have an answer for that. I mean, in terms of sensitivity, in this sense, I think what we're going to have to look at is the concentration. How much lactate, C-13 level lactate, do we need in the tissue? What micromolar, what nanomolar can we detect? In terms of sensitivity, let's say if you want to, how much Warburg effect do we need in order to see that? I think, I don't have an answer to that, but I think it's more like, if it's enough for you to have, let's say, 1 millimolar of lactate, you can see it. If it's less than that, you may not. But that's a number I just threw around there to make my point.
So you'd have to just empirically kind of.
Yeah. The field is still pretty new. And I mean, there's a few sites who have done this in human patients. That's something we're trying to do here too. But we don't have the polarizer, we don't have the setup yet, but we need to have the money.
So maybe the last question, unless somebody else has one. On all of these, what are the limitations for formulating in vivo versus ex vivo, meaning where you have to take out the organ that versus real life studies on animals or humans?
Actually, we can do an animal studies here, like in vivo, here and at UT Southwestern that I was at. And we did have some. I just didn't include it. But we have everything in terms of what is needed to do that. And the question, it depends on what we are interested in, I guess. Yeah.
All right. Well, thank you so much.
Well, thank you very much.